If you are captivated by fascinating world of microbiology and thirst for knowledge, subscribing to our channel is an absolute must. Join us on this incredible journey as we delve into the microscopic wonders of bacteria and unravel the secrets of microbial world. With each captivating video, you will gain a deeper understanding of intricacies of microbiology. Don't miss the opportunity to expand your scientific horizons and satisfy your curiosity. Hit that subscribe button now and embark on an eye-inspiring adventure into the captivating realm of microbiology. Cultivation of microorganisms. Cultivation refers to growing and propagating organisms such as bacteria, fungi, or plants by creating and maintaining the necessary environmental conditions for their growth. These conditions are crucial for organisms to survive, reproduce, and thrive. Several key factors need to be considered and controlled when cultivating bacteria to ensure their successful growth. These factors are nutrients, which include carbon, nitrogen, sulfur, phosphorus, mineral, pH, temperature, aeration, salt concentration, and ionic strength of the medium. Now we will explain these factors step by step. Nutrients. Bacteria require specific nutrients to grow, including carbon, nitrogen, sulfur, phosphorus, minerals, and other essential elements. These nutrients serve as the building blocks for cellular structure, energy production, and various metabolic processes. pH. The medium's pH level, which measures the acidity or alkalinity, play a significant role in bacterial growth. Different bacteria have different pH preferences, but a pH range of 7.2 to 7.6 is generally considered optimal for many bacterial structures. Temperature. Bacteria exhibit a wide range of temperature requirements for growth. Some bacteria thrive in moderate temperatures, while others prefer extreme heat or cold. Controlling the temperature within the appropriate range is crucial for promoting bacterial growth. Aeration. Oxygen availability is essential for the growth of aerobic bacteria that require oxygen for their metabolic processes. Proper aeration ensures a sufficient oxygen supply to support their growth. However, some bacteria are anaerobic and can survive without the oxygen. Salt concentration. Some bacteria are halophiles, which mean that they require a certain level of salt concentration for growth. Adjusting the salt concentration in the growth medium is necessary to create a suitable environment for these organisms. Ionic strength. The ionic strength of medium refers to the concentration of ions in the growth medium. It influences the osmotic balance within the bacterial cells and affects the growth and metabolism. Maintaining an appropriate ionic strength is essential for the bacteria's overall health and growth. By carefully manipulating and optimizing these environmental factors, scientists and researchers can create conditions that support the growth of the specific bacteria in laboratory settings. Cultivating bacteria in controlled environment enables scientists to study their characteristics, behaviors, and potential applications in the various fields such as medicine, biotechnology, and environmental science. This is the picture of different culture medias. Now we will discuss the preparation of culture media. Culture medium is made of broth or extracts of animal meat. Some other materials for bacterial growth support is added to that medium. The base of many culture media is derived from animal sources, such as meat extract or infusion. These materials contain various nutrients that serve as a source of carbon, nitrogen, vitamins, and other essential compounds required for bacterial growth. Peptone. Peptone is commonly used ingredient in the culture media. It is produced by partially digesting proteins through enzymatic hydrolysis. Peptone provides a rich source of amino acids and peptides that are essential building blocks for bacterial proteins and other cellular components. Salt. Salt in sodium chloride is often added to culture media to maintain the osmotic balance necessary for bacterial growth. It helps to create an optimal environment for the bacteria by controlling water movement across the cell membranes. Serum, usually obtained from the blood, contains a wide range of growth factors, that is hormones and other compounds that support the growth and viability of cellular bacteria. It provides additional nutrients and enhances the culture medium's ability to sustain specific bacterial species. Blood. Blood is sometimes used as an ingredient in specialized culture media, particularly cultivating fastidious bacteria that require specific growth factors from blood component. Blood agar, for example, is a medium containing blood and agar commonly used to culture bacteria that require additional nutrients or exhibit particular growth characteristics. Autoclave. Autoclaving is sterilization technique that involves subjecting the culture media to moist heat at a high temperature, usually around 121 degrees centigrade for specific duration for 15 to 30 minutes. Autoclaving destroys or inactivates any existing microorganisms, ensuring that the culture medium is free from contaminants and provides sterile environment for bacterial growth. By combining these ingredients and subjecting the culture media to autoclaving, scientists create a nutrient-rich, sterile, and supportive environment for bacterial growth in the laboratory. The specific composition of the culture media can be tailored to meet the nutritional requirement of different bacterial species, allowing researchers to cultivate and study specific microorganisms selectively. What are the different types of culture media? According to consistency, the culture media are solid medium, semi-solid medium, and liquid medium. According to nutrition, basic medium, enriched medium, selective medium, differential medium or indicator medium, and transport medium. Solid culture media. Solid media plays a crucial role when it comes to culturing bacteria or other microorganisms in a laboratory setting. Solid media are prepared by incorporating a gelling agent such as agar or gelatin into the culture medium. This gelling agent solidifies the medium, providing a solid surface for bacterial growth. Solid media are commonly used in petri dishes, which are shallow, flat, round plates with a lid. These petri dishes are filled with the solid medium and used for plate cultures. In plate cultures, a small amount of sample containing bacteria is spread evenly on the surface of the solid medium. As bacteria grow, they form distinct and isolated colonies on the solid surface, which are visible to the naked eye. Each colony represents a cluster of cells derived from a single bacterium. Solid media can also be used in bottles or tubes for different cultures. In step cultures, a sterile needle or loop is used to introduce the bacteria into solid medium by stepping it into the medium. This creates a vertical column of bacterial growth. Slope cultures, conversely, involve streaking the bacteria in zigzag pattern on the slanted surface of the solid medium in the tube or bottle. The primary purpose of the culturing bacteria on a solid medium is to isolate discrete colonies of each organisms present in the specimen. By providing a solid surface, the medium allows for the separation and growth of individual bacteria, making it easier to observe and study them. Each colony represents the single species or strain of bacteria, enabling microbiologists to identify and analyze different types of microorganisms present in a sample. Isolation of discrete colonies is essential for further characterization, identification, and analysis of bacteria. It helps researchers studying morphology, growth characteristics, and biochemical properties of individual bacteria strains. Additionally, the isolated colonies can further cultured or subjected to various tests and experiments to determine their specific traits and behaviors. Overall, the solid media provide a valuable tool for microbiologists to isolate, observe, and study individual bacteria, facilitating the understanding of microbial diversity, behavior, and potential application in various fields of research and industry. Semi-solid media are a type of culture medium that has a gel-like consistency, intermediate between solid and liquid media. They are prepared by adding a small amount of agar, usually around 0.4 to 0.5% weight by volume, to a liquid medium. The addition of agar to the liquid medium allows it to partially solidify, creating a semi-solid texture. This consistency is achieved because agar, a gelatinous subject derived from a seaweed, forms a network of interconnected fibers when heated and then solidifies as it cools. The concentration of agar used in semi-solid media is lower compared to a solid media, resulting in a softer gel-like texture. Semi-solid media serve various purposes in the morphology: transport media, motility tests, and biochemical tests. Transport media, semi-solid media are commonly used as a transport media for preservation and transportation of bacteria from one location to another. The semi-solid consistency helps to immobilize the bacteria within the medium, preventing their movement during transit. It also helps to maintain the viability and integrity of bacteria until they reach the laboratory for further analysis. Motility tests. Bacterial motility refers to the ability of bacteria to move and exhibit self-propelled motion. Semi-solid media are frequently employed to assess bacterial motility. When a bacterium is motile, it can actively swim or move through the semi-solid medium, resulting in visible spreading or growth away from the point of inoculation. Non-motile bacteria, on the other hand, do not exhibit such movement. Biochemical tests. Semi-solid media are also utilized in the biochemical test to assess metabolic capabilities of bacteria. These tests often involve the inoculation of semi-solid medium with a specific substrate or indicators. The growth and diffusion pattern of bacteria within the semi-solid medium can indicate the presence or absence of certain metabolic activities, such as ability to ferment specific sugars or produce enzymes. Due to their gel-like consistency, semi-solid medium provide a controlled environment for assessing bacterial motility and performing biochemical tests. They allow for observable movement or spreading of motile bacteria and provide a suitable substrate for biochemical reactions to occur. The versatility of the semi-solid media make them valuable tools in microbiology laboratory, particularly for transport purposes and for assessing bacterial motility and biochemical characteristics. Liquid culture media, as the name suggests, are the culture media that exist in a liquid or fluid form. They are widely used in various applications within the microbiology. Liquid media are commonly employed as enrichment media when the target organisms are expected to be present in the low numbers or when the sample contains complex mixture of microorganisms. Enrichment media provide favorable conditions to enhance the growth of specific bacteria, allowing them to proliferate and outcompete other organisms present in the sample. A classic example is the use of liquid blood culture media to detect the presence of bacteria in the blood samples, as bacteria may be present in very low numbers. Certain microorganisms such as Vibrio cholera exhibit a distinctive growth pattern where they produce a visible film or surface growth on the liquid medium in which they are cultured. This growth can be observed in a specific conditions, such as when Vibrio cholera is grown in alkaline peptone water. The formation of surface growth can be indicative of specific characteristics or behaviors of the organisms. Liquid media are frequently employed for performing biochemical tests on microorganisms. These tests assess the metabolic capabilities of the bacteria, including their ability to ferment specific substrates or produce particular enzymes. The liquid medium provides a homogeneous environment for conducting these tests, allowing for standardized and reproducible results. Liquid culture media offers several advantages in the microbiology. They provide a uniform and easily mixed medium, ensuring equal distribution of nutrients and promoting unrestricted bacterial growth. Liquid media also facilitate the scalability of the bacterial cultures, allowing for the growth of large quantities of microorganisms in bioreactors and fermentation process. Liquid media provide a suitable environment for biochemical reactions and facilitate the addition of specific nutrients or growth factor. This flexibility makes them particularly useful in application such as enrichment, surface growth studies, and biochemical testing. This is the solid medium, this is the semi-solid medium, and this is the liquid medium. Now we will discuss the types of culture media according to the nutrition. Number one is basic media. Basic media, also known as the general or non-selective media, are simple culture media that provide the essential nutrients required for the growth of a wide range of microorganisms. These media, such as nutrient agar and nutrient broth, are formulated to support the growth of microorganisms that do not have a specific or specialized nutritional requirements. Basic media contain a variety of ingredients that provide the necessary carbon and nitrogen sources, minerals, vitamins, and water to support bacterial growth. They are typically composed of peptone, partially digested proteins, beef or yeast extracts, and agar in the case of agar-based solid media. These components provide a range of nutrients that can meet the basic requirements of many organisms. The use of basic media allows for the cultivation of variety of common bacteria. Some examples of the bacteria that can grow on basic media include E. coli, Staph aureus, Pseudomonas, and Proteus. Enriched media. Basic media can be enriched with various components to provide additional nutrients and growth factors that support the growth of wide range of microorganisms. Enriched medium are designed to provide specific nutritional requirements that may be necessary for the growth of certain organisms, particularly fastidious organisms that have specialized nutritional needs. Enrichment of basic media involves the addition of substances such as whole or lysed blood, serum, peptones, yeast extracts, vitamins, and other growth factors. These components supply essential nutrients, growth promoting factors, and specific metabolic substrates that may be required by the certain microorganisms. The purpose of using enriched media to increase the number of particular pathogens by providing all the necessary ingredients to support its growth. By meeting the specific nutritional requirements of the target pathogens, an enriched medium creates optimal condition for its proliferation and allows for the selective growth of desired organisms. Enriched media are particularly crucial for the cultivation of fastidious organisms that have specific nutritional needs. Fastidious organisms are those that require particular growth factors or nutrients that are not present in the basic media. Examples of such organisms include H. influenza, Neisseria species, and certain Streptococcus species. These organisms often have complex nutritional requirements that necessitate the use of enriched media to support their growth. Two commonly used enriched media are blood agar and chocolate agar. Blood agar is a type of enriched medium where whole blood, typically sheep or horse blood, is added to the agar base. This addition provides additional nutrients such as red blood cell components that support the growth of wide range of microorganisms including pathogenic bacteria. Chocolate agar on the other hand is made by heating and lysing the blood added to the agar base, releasing additional nutrients and growth factors that facilitate the growth of fastidious organisms. Enriched media play a critical role in isolation, cultivation, and identification of specific microorganisms with a specialized nutritional requirement. By providing the necessary nutrients and growth factors, enriched media enable researchers to successfully culture and study a wide range of bacteria including those that are challenging to grow on basic media alone. Selective media. Selective media are the solid media that contain substances such as bile salts, chemicals, dyes, or antibiotics that inhibit the growth of certain organisms while allowing the growth of others. These substances create an environment that selectively favors the growth of one organism over the others, making it easier to confirm the presence of desired organisms. Selective organisms are particularly useful when culturing specimens from sites that have a normal microbial flora, where there is a risk of unwanted contaminants overgrowing the target pathogens. By inhibiting the growth of normal flora or other contaminants, selective media help isolate and identify the specific pathogens of the interest. For example, Vibrio cholera, a bacterium that is responsible for cholera, can be isolated using a selective medium such as TCBS agar. TCBS agar contains bile salts and other selective agents that inhibit the growth of many bacteria while allowing Vibrio cholera to grow and produce distinctive colonies. Temperature can also be used as a selective factor. Certain organisms have specific temperature requirement for growth. For example, some bacteria can grow at low temperatures such as 4 degrees centigrade, while many other organisms are inhibited or grow poorly at such temperatures. This temperature selectively can be used by culturing specimens on media incubated at specific temperatures to favor the growth of Listeria over other bacteria. Selective media can be designed to target specific group of organisms or type of pathogens. For example, TSI triple sugar iron agar is slanted tube medium that contains sugar, iron salts, pH indicators. It is commonly used for the isolation and differentiation of enteric bacteria, including those that can ferment sugar and produce characteristic color change in the medium. Another example is eosin methylene blue EMB agar, which is an enteric isolation medium. EMB agar can contain dyes that inhibit the growth of gram-positive bacteria while allowing the growth of gram-negative bacteria, particularly enteric organisms such as E. coli and Salmonella. EMB agar can differentiate between different enteric organisms based on their ability to ferment lactose and produce characteristic metallic green and dark purple colonies. Selective media play a crucial role in microbiology by selectively inhibiting the growth of certain organisms while allowing the growth of others, facilitating the isolation and identification of specific pathogens or desired organisms for complex microbial structure. These media use various substances, chemicals, or temperature conditions to create a selective environment that favors the growth of the target organisms while suppressing the growth of unwanted contaminants. Indicator media or differential media. Differential media are the culture media that contain dyes or other substances that aid in differentiating the microorganisms based on specific characteristics or metabolic activities. These media are designed to produce distinguishable changes in appearance or color, allowing for the differentiation of the different bacterial species or identification of specific metabolic capabilities. One common approach in differential media is the use of indicators that change color in response to certain metabolic activities. For example, many differential media incorporate indicators that change color in the presence of acid, which is produced as a result of fermentation of specific carbohydrates by bacteria. When a bacteria ferments a particular substance, that is carbohydrate, such as lactose or glucose, it produces acids as a byproduct. The indicator in the medium will change color if acid is produced, enabling the differentiation of the bacteria based on their ability to ferment specific carbohydrates. As an example of differential media is the McConkey agar. McConkey agar contains bile salts, crystal violet dye, lactose, a pH indicator. It is commonly used to differentiate between lactose fermenters and non-fermenters among gram-negative bacteria, particularly those from the family Enterobacteriaceae. Lactose fermenting bacteria produce acid during fermentation, causing the pH indicator to turn red or pink. Non-fermenters, on the other hand, do not produce acid and the medium remains pale or colorless. It's worth noting that many media used to isolate pathogens or specific bacteria serve multiple purposes, combining both selective and differential properties. For example, a medium may be both selective, inhibiting the growth of unwanted organisms, and differential, allowing the differentiation of specific bacterial species based on their metabolic activities or other characteristics. Transport media. Transport media are specialized semi-solid or liquid media used to preserve and maintain the viability of microbial specimens during the transportation from the collection site to laboratory. These media are particularly important when specimens cannot be immediately cultured after collection, as they help prevent the overgrowth of commensal organisms while ensuring the survival of both aerobic and anaerobic pathogens. The primary purpose of transport media is to maintain the integrity and viability of the pathogens present in the specimen until they can be processed and cultured in the laboratory. They contain specific ingredients that provide a suitable environment to sustain the microorganisms, preventing their death or loss of viability during transit. Transport media finds significant application in the transportation of biological specimen from health centers to district microbiology laboratories or various collection sites to regional public health laboratories. They play a crucial role in preserving the integrity and viability of pathogens, enabling accurate and reliable testing and identification in the laboratory. Examples of the commonly used transport media are Carry-Blair medium. This medium is specifically designed for preserving enteric pathogens such as bacteria that cause gastrointestinal infection. It contains a combination of buffering agents, reducing agents, and antimicrobial agents that help maintain viability of enteric pathogens during transportation. Image transport medium. Image medium is used to ensure the viability of specific pathogens such as Neisseria, the causative agent of gonorrhea. It typically contains a gel-like matrix, salts, and nutrients that provide a favorable environment for the survival of the pathogen during transit. Thank you from the depths of our hearts for gracing us with your presence. Until we meet again, may your curiosity be even ignited and your thirst for knowledge be unquenchable. Thank you, dear viewer, for being an integral part of our remarkable community.